Hello, in this video, we are going to explain about glycolysis, Krebs cycle, ring reaction, and electron transport chain. Glycolysis is the first step in the breakdown of glucose to extract energy for cellular metabolism. Glycolysis consists of an energy-requiring phase followed by an energy-releasing phase. Glycolysis takes place in the cytosol of a cell. Phosphorylation of glucose catalyzed by hexokinase to form glucose 6-phosphate by ATP. Glucose 6-phosphate rearranged into fructose 6-phosphate. Phosphorylation of fructose 6-phosphate catalyzed by phosphofructokinase to form fructose 1,6-bisphosphate by ATP. Fructose 1,6-bisphosphate split into dihydroxyacetone phosphate DHAP and glyceraldehyde 3-phosphate G3P. DHAP rearranged to form G3P. 2G3P oxidized and phosphorylated to form 1,3-bisphosphoglycerate. NAD+ reduced to NADH and H+. 1,3-bisphosphoglycerate undergoes substrate-level phosphorylation to form 3-phosphoglycerate and gain 1 phosphate group. 3-phosphoglycerate rearranged into 2-phosphoglycerate. 2-phosphoglycerate dehydrated and form phosphoenol pyruvate. In this process, H2O being released. Phosphoenol pyruvate undergoes substrate-level phosphorylation to form pyruvate and gain 1 phosphate group. Here is a full process of glycolysis. These are the product of glycolysis. Now, let us look at link reaction. Link reaction occur in mitochondrial matrix. It involves 3 reactions. A carboxyl group from pyruvate is removed as carbon dioxide and form a 2C molecule. The remaining 2C molecule is oxidized by transferring their electron and H+ to NAD+. Acetate combines with coenzyme A to form acetyl-CoA, which then enters the Krebs cycle. These are the product of link reaction. Now, let us look at Krebs cycle. Krebs cycle occur in mitochondrial matrix, involve 8 reactions and each catalyzed by specific enzyme. Acetyl-CoA combines to oxaloacetate, forming citrate. Coenzyme A is released and free to combine with another 2 carbon group to reform acetyl-CoA. Citrate undergoes isomerization and is converted to its isomer, isocitrate. Isocitrate is converted to alpha-ketoglutarate through oxidative decarboxylation. Carbon dioxide and NADH are produced. Alpha-ketoglutarate is converted to succinyl-CoA through oxidative decarboxylation. Carbon dioxide and NADH are also produced. Succinyl-CoA is converted to succinate. Phosphate group is transferred to GDP, forming GTP and later form ATP. Succinate is oxidized to fumarate. Hydrogens are transferred to FAD. FAD is reduced to FADH2. Fumarate will react with water to form malate. Malate is oxidized to oxaloacetate. Hydrogen is transferred to NAD+. NAD+ is reduced to NADH. This is the full step of Krebs cycle. These are the product of Krebs cycle. Now, let us move to electron transport chain. Electron transport chain occur in inner membrane of mitochondria. Complex 1 oxidize NADH to NAD+, then complex 1 transfer electron to ubiquinone. Complex 2 oxidize FADH2 to FAD, then complex 2 transfer electron to ubiquinone. Ubiquinone accepts electron from complex 1 and transfer to complex 3. Ubiquinone accepts electron from complex 2 and transfer to complex 3. Complex 3 accept electron from reduced ubiquinone. Then, complex 3 transfer electron to cytochrome C. Cytochrome C accepts electron from complex 3 and transfer to complex 4. Complex 4 accepts electron from cytochrome C and transfer electron to oxygen. 2H+ combine with 2 electron and half oxygen to form water. Electron transport chain does not generate ATP directly, so chemiosmosis is needed. H+ pass from intermembrane space to mitochondrial matrix through the channel of ATP synthase. This activates ATP synthase and it catalyzes the formation of ATP from ADP and inorganic phosphate. These are the product of electron transport chain. Thank you for watching. Bye.